أعزائي الطلاب، السلام عليكم ورحمة الله. راح نبدأ التسجيل الصوتي لشرح محاضرة التيومر immunology. طبعا content of the lecture will be about the tumor antigens, which are the tumor markers, both tumor associated and tumor specific antigens. Also we will review the immune response to tumors, and we will talk about the evasion mechanism of the tumor by the immunity. I mean the immune evasion mechanism of the tumor. Also the cancer immunotherapy techniques. We will talk about it. The tumor antigens. As you know, every tumor have specific antigens, and these antigens are of two types: either tumor specific antigens or they are tumor associated antigens. The tumor specific antigens necessitate the presence of mutation. So when there's a mutation of the certain cellular gene, it will produce a tumor specific antigen. We have two types of tumor specific antigens leading to this mutation: either chemically induced mutation or virally induced mutation. And by its name, tumor specific antigen are specific for the antigen. That's to say, they are not present unless there is a tumor source. The second type of tumor antigen are the tumor associated antigens, and these are normal cellular proteins, so there is no mutation. They are normal cellular proteins, but they are aberrantly expressed, overexpressed. Some examples are oncofetal tumor antigen, such as alpha fetoprotein and human chorionic gonadotrophin, and also we have the oncogene proteins, and also some hormones and hormone receptors, growth receptors, certain antibodies. All of these are regarded as tumor associated antigens because they are overexpressed. They are normal cellular products that are overexpressed. We'll talk in more detail about the tumor antigens and tumor markers during the laboratory part. This figure shows the tumor specific and the tumor associated antigen, and to explain what we mentioned about the two types of tumor markers. In case of tumor specific antigens, they are neoantigens and are expressed in cell but not on normal cells, so they are only expressed in tumor cells. Usually there is presentation of mutant peptide from mutated cellular protein, as we mentioned, and they are called tumor specific antigen. The tumor associated antigen are of two types: either reactivation of embryonic genes, so normal embryonic genes are reactivated in adulthood, or overexpression of normal self-protein by tumor cell. So these are normal cell peptide, such as hormones, as we said. The other figure entitled tumor antigens is explaining certain examples of the tumor antigen, and these antigens are classified according to the types. First, the differentiation type or class, and this might be an example of this type is tyrosinase and surface immunoglobulin. And also we have abnormal gene expression such as HER2 receptors in breast cancer and ovarian cancer, which is tyrosine kinase receptor, as regarded the growth factor receptor. Other class is the abnormal post-translation modification, which is the MUC1 protein of the breast and pancreatic cancer. We have the fourth class of tumor antigens, oncoviral proteins, such as human papilloma virus 16, E6 and E7 proteins. These are present in the cervical carcinoma. Here another table about other types or examples of the viral associated human cancer, viruses associated with human cancers. We have several types and maybe repeated here: the papilloma virus, which causes the wart and carcinoma of the cervix. We have the hepatitis B virus, which might cause liver cancer, hepatocellular carcinoma. Epstein-Barr virus could lead to Burkitt's lymphoma or nasopharyngeal carcinoma, less commonly, or B-cell lymphoproliferative disease. It usually lead to Burkitt lymphoma. We have, these are the DNA viruses. We have the RNA viruses, which are human T-cell leukemia, leading to a rare tumor of the T-cell leukemia. We have another virus, RNA virus, which is the HIV virus, leading to Kaposi sarcoma. It's also a tumor associated with HIV and occur in AIDS patients in the later stages. The last two tables we have only examples of some viral specific antigen associated tumor with human cancer, and it may be kept in mind. Here we have chromosomal translocation. This is tumor specific, an example of a tumor specific antigen, which, as we said, is represented by a certain mutation leading to a genetic disruption, and this genetic disruption is specific for that tumor. Here the example for the CML, chronic myeloid leukemia, with the Philadelphia chromosome, which is the backbone of the diagnosis of the disease. Philadelphia chromosome is a translocation between chromosome 9 and chromosome 22, and we have this figure shown by the karyotyping assay, as we will see in the laboratory of the tumor markers. The other tumor-specific antigen or tumor-specific gene, which is the occurring Burkitt lymphoma, we have the c-MYC gene, and the c-MYC gene is a sort of chromosomal translocation between chromosome number 8 and 14, and this figure shows this translocation. And when we see this translocation, which is the c-MYC, referred to as c-MYC, this means that this sample belongs to a patient with Burkitt lymphoma. So that's why it's called tumor-specific antigen. So this type of translocation is specific for Burkitt lymphoma, and the previous one, translocation between 9 and 22, which is Philadelphia chromosome, is specific for the CML. Now, after we mention some points about tumor markers or tumor antigen, we will review the immune response against tumors. As you know, the immune system is responsible for defense against pathogen and tumors. So we'll talk about the immune response against tumor. We have the immune surveillance theory, and we have certain point about some immune cells specific against tumor. And this figure shows the general concept of immune response against tumor, talking about broadly about the humoral and cell-mediated arms of the immune response, and starting by the cell-mediated immunity represented by the cytotoxic T lymphocytes. The cytotoxic T lymphocyte react and respond and bind to the tumor antigen presented by antigen presenting cell. So the tumor have antigen, and these antigens are ingested by antigen presenting cell. It may be dendritic cell or the tumor cell itself. This antigen is presented in association with MHC class 1 to the cytotoxic T lymphocyte, and then the cytotoxic T lymphocyte activated and divided into many cells. Some cells are regarded as memory cells, other cells are regarded as activator cells and or effector cells, and these cells will kill the tumor cells. The other part of the cell-mediated immunity is represented by the T helper lymphocyte, which is the CD4 positive lymphocyte. The first one was CD8 positive, which is cytotoxic T lymphocyte, as you know. And the second type is the CD4 lymphocyte, which are the helper lymphocyte. Again, the helper lymphocyte, CD4 positive lymphocyte, recognize and bind the tumor antigen presented by the antigen presenting cells, which are only the macrophage, dendritic cell, and B lymphocytes. T helper 1 receive antigen from macrophage and dendritic cell, as you know, and this binding with the antigen bound to the MHC2 lead to activation of the T helper 1. Activation of T helper 1 also again lead to division of the T helper 1 cell, giving rise to many large number of T helper lymphocytes. Some sets are kept for the future as memory cell. Other sets are become effector. They will help the cytotoxic T lymphocyte. And regarding T helper 2, they will help the B lymphocytes. So the helper lymphocyte acts both on the humoral and cellular response of the immunity against tumor by helping cytotoxic T lymphocyte and helping the B lymphocyte, which are the cells responsible for the antibody production, i.e. responsible for the humoral response. Also, the T helper 1 and T helper 2 secrete some cytokines. These cytokines acts against the tumor. We have the B lymphocyte, which is responsible for antibody production either by itself or by the help of T helper 2. In both cases, B cell will produce antibodies, and we will show the role of antibody here. And these antibodies represent the humoral immune response. Again, the B lymphocyte, when activated, give rise to large number of cells. Some of these cells are kept as memory B lymphocytes for the future attack with the contact with the tumor. The other slide talking about the professional antigen presenting cell, and this is a review. You have already studied the antigen presenting and antigen presentation process. So we have mainly three types of professional antigen presenting cell, and broadly speaking, we have professional antigen presenting cell and general antigen presenting cell. The general antigen presenting cell could be any cell in the body infected by virus or being a tumor, transformed into tumor. The professional antigen presenting cell is studied here in this slide are only three types of cells: dendritic cells, macrophages, and B cells. The dendritic cells and macrophages represent professional antigen presenting cell presenting antigen into T helper 1 lymphocyte mainly, and also the B lymphocyte are antigen presenting cell presenting the antigens mainly to the T helper 2, as you see in the previous figure. So all types will internalize the antigen and process it and presented parts of the antigen with the MHC for the dendritic and macrophage with MHC class 2, also for the lymphocyte for MHC class 2. All these antigens bound to the MHC class 2 will be presented to the corresponding T helper lymphocyte, leading to the activation of the APC itself, activation of the dendritic cell and macrophage and B cells, and also activation of the T helper, which will receive the and bind to the antigen. The activation of this antigen presenting cell to the T helper cell need many signals. We will show these signals. One of the signals are binding of the antibody with the binding of the T helper T cell receptor with the antigen and MHC complex. The other signal is the co-stimulatory signal, and the third signal is the cytokine production. For the dendritic cell, as antigen presenting cell, dendritic cell in case of a tumor immunity can present antigen both to the CD4 T lymphocyte and CD8 T lymphocyte. That's to say, for the T helper lymphocyte and T cytotoxic. So dendritic cell can express both MHC1 and MHC2. So, by continuing speaking about the priming of T lymphocyte via the antigen presenting cell, we have many signals, or we have exactly three signals. First is the binding between the T cell receptor and the antigen peptide combined to the MHC molecule. Second signal is the co-stimulatory signal, which is binding of CD28 on the T cell with CD80 and CD86 on the dendritic cell. The third, if this first and second signal are present, the third signal will exist, which is activation and an inflammatory cytokine production. So this is the full picture of priming of T lymphocyte, having three signals. First, binding of the T cell receptor with the MHC antigen peptide complex. Second, binding of CD28 with the CD86 on the dendritic. The third signal is follow only when the first two signals are present, which is the inflammatory cytokine production and activation states, or priming states. The other slide also further explain this process of priming of T cell by the antigen presenting cell As we mentioned, all of these figures are review for the process of antigen presentation and activation of T lymphocyte. So we have here T lymphocyte and primed by the antigen presenting cell. Here we have the signal 1, which is antigen binding between specific binding between T cell receptor and MHC class 2 with the antigen peptide bind to it. The second signal is the co-stimulatory signal between CD28 and CD8086, or CD40 ligand with the CD40 of the antigen presenting cell. The third signal is cytokine production. Talking about the natural killer cell. The natural killer cell is part of the immune surveillance mechanism against the tumor, as you know. Beside this cytotoxic T lymphocyte, natural killer cell is part of the innate immunity. It acts by killing of the tumor cell and virus infected cell, and this totally depends on the presence of amount of normal amount and shape of MHC class 1 on the cell. Any cell, if it is normal cell, have certain amount and certain shape of MHC class 1 known by the natural killer cell. If the MHC class 1 on the body cell is abnormal or absent, the natural killer cell have killer activation receptor, which is activated and lead to killing of this abnormal cell, and this is termed missing cells. Some transformed cell, malignant cell, may keep the normal MHC class 1 but express more killer activation receptors. Also, this case or this condition is called induced self, and this also lead to killing of the tumor cell by the natural killer cell. So we have two cases of killing activation: if the self is missing, or if induced self by the presence of MHC1 plus the killer activation receptor. The process by which the natural killer cell kill the tumor cell is by releasing the granzyme and cytotoxin, which lead to cytolysis of the tumor cell. And this is also a revision for the previous study about the natural killer cell killing mechanism. The other slide about natural killer cell killing mechanism is further explain the process. On the left side we have a normal cell having killer inhibitory receptors represented by normal MHC1 class, while the figure on the right represents abnormal cell lacking MHC class 1 molecule, i.e. lacking killer inhibitory receptor, so it will have more killer activation receptor and lead to killing of this abnormal cell by the natural killer cell. Now the other slide talk about the antibody mechanism of action. Mechanism of action of antibody against tumor. What is the benefit of antibody? What is the role of the humoral immune response here? Antibody represent the humoral arm of the immunity. As you know, antibody is secreted by the activated B lymphocyte against certain tumor. Antibody have several mechanisms to act against tumor. The first mechanism is shown in this figure by activation of complement via classical complement pathway of activation. And as you know, complement activation lead to production of many complement product leading to an inflammation and an aflatoxin production, and also cell lysis. The other mechanism of the antibody is by binding of this antibody with the, so we have antibody fixed to the tumor surface cell, and this antibody will be bound or captured by a macrophage, as you know, or sorry, natural killer cell, or even macrophage. The macrophage have a receptor for the antibody. It's called Fc gamma receptor, and these receptors will lead the macrophage to capture the antibody, and by capturing the antibody, the macrophage will activate other cells like natural killer cells. The other slide will continue the mechanism of antibody action against the tumor, and here we have important mechanism of the antibody, which is called antibody dependent cell mediated cytotoxicity. This is shown on the left. When the antibody bound to the specific tumor cell, again the antibody have a receptor on the natural killer cell, so this natural killer cell will be adhere to the and bound to the tumor cell via the binding of this antibody with the Fc receptor antibody on the cell surface of the natural killer cell. This will lead to the activation of the natural killer cell by different way that mentioned about MHC. This is non-MHC dependent. This is depend only on the antibody binding, and this may take another name, which is called antibody dependent cell mediated cytotoxicity by the natural killer cell, and it might belong to the adaptive arm of the immune response, because there is antibody in the pathway of killing. So when the natural killer cell bind to the antibody bound to the target cell, tumor cell, this will activate the natural killer cell killing activity, releasing the perforin and the granzymes, and lead to lysis of the tumor cell. And this process is called antibody dependent cell mediated cytotoxicity. On the same figure on the right as field of the figure, we have macrophage. Again, the macrophage have a receptor for the antibody called Fc receptors, and we have tumor cells surrounded by antibodies, specific antibodies. The macrophage will bound to the antibodies, and this lead the macrophage to form pseudopodia and phagocytose the tumor cell. So this is called antibody dependent cellular phagocytosis, and this is again another mechanism of action of antibody against tumor cells led by the macrophage. The next slide shows the cycle of cancer immunity. We have, as a summary of the cancer immune response, this cycle, and this is just representation of the cycle in a diagram, starting by step 1, which is the release of the cancer cell antigen resulting from cancer cell death. These antigens are presented by the dendritic cell, and dendritic cell will prime the T cell and activate the T cell. T cell will act against the tumor cell by killing, and this is done by cytotoxic T lymphocytes after they infiltrate the tumor and recognize the tumor antigen, and finally kill the cancer cell. So the first step is release the antigen. Second step, the cancer antigen captured by the antigen presenting cells. Third step, antigen presenting cell prime the T cell, mainly the cytotoxic T lymphocyte, which migrate via the bloodstream and lymph node and infiltrate the tumor in the fifth step. In the sixth step, these primed cytotoxic T lymphocyte recognize tumor cell antigen and kill them in the final and then in the last step. And this is, as you see, continuous cycle and a dynamic cycle, and there is a continuous battle between the immunity and the cancer. The other figure denoted by 3 E's also speak about this cycle in another way. We have 3 E, which are elimination, equilibrium, escape, and these conditions are representing a battle between immunity and the cancer, and this battle may end by the death of the cancer on the left side, elimination, or might end with the escape of the cancer and loss of the immune system on the right side, or the battle may be have a state of equilibrium in the middle, and there is continuous battle with the presence of both the tumor and immune cells. Any patient with cancer may have any of these results of the battle between immunity and the tumor. Now another subject, which is the tumor evasion. How the tumor can evade and escape from the immune response. We have many mechanisms by which tumor can escape the immune system. The first mechanism is immunological enhancement or exhaustion by antigen shedding. This means some tumors continuously shed antigens, and these antigens are non-protective, and they are only responsible for exhaustion of the immune system, and thus the tumor will grow away from the exhausted immune system. This one mechanism. Another mechanism is modulation of the tumor antigens. Some tumors modulate their antigens and change their antigens, and this change will lead to misleading of the immune cells. Another mechanism is some tumors reduce or underexpress MHC class 1. As you know, MHC class 1 is an important mean of killing of the tumor, both by natural killer cell and cytotoxic T lymphocyte. So when the tumor underexpress its MHC class 1, it will evade the killing of by the cytotoxic T lymphocyte. Some tumor cells produce no co-stimulatory signal or inhibit the co-stimulatory signal, which is the signal number 2, as we mentioned in the priming of the T lymphocyte. The signal number 2 is inhibited by the presence of two molecules, PD and CTLA4, or two pathways. We have two pathways to inhibit the co-stimulatory signal, either by PD or by CTLA4 pathway. Both pathways lead to inhibition or absence of co-stimulatory signal. This means that these pathways lead to prevention of priming of T lymphocyte. This phrase is translated by the immunity into another term called energy. So we have energy of the T lymphocyte, energy written by A, energy mean inhibition of the T lymphocyte. The energy is used by some tumors, and we have two pathways, two main pathways by which energy is fixed, and these two pathways are the PD and CTLA4 pathways. This figure in the slide 22 explain how the tumor escape the immunity by decreasing the expression of MHC class 1. As you see here, if the tumor cell have low expression of MHC class 1, they will escape killing by the cytotoxic T lymphocyte, while if they have high expression of MHC class 1, they will killed rapidly and readily by the cytotoxic lymphocyte, as it is clear here. And the other slide show the mechanism by energy, which is inhibition of the co-stimulatory signal by the tumor. And this energy is referred to another broader term called peripheral tolerance. I think you study the peripheral tolerance in case of autoimmunity. So the peripheral tolerance is a mechanism by which the body overcome autoimmunity. This peripheral tolerance or energy is used by some tumor to escape from the immunity. And as we mentioned, we have two main pathways to develop energy. These two pathways are CTLA4 pathway and PD1 pathway. Both pathways aim to inhibit the co-stimulatory signal. This means they will lead to inhibition of the priming of the T lymphocyte, or they will cause energy of the T lymphocyte. In the first case, we have dendritic cell presenting antigen into the T lymphocyte. We have first signal binding of the T receptor with the peptide and MHC. Second signal is the co-stimulatory signal CD28 on the T cell and CD80 on the dendritic. This is normal priming. If there is CTLA4 on the right, this will prevent priming and lead to energy. This CTLA4 is produced by the tumor cell. Again, in the section B of the slide, we have dendritic cell presenting antigen to the T lymphocyte, and we have binding, which is the first signal, and we have the second signal absent and replaced by PD and PD ligand binding of PD and PD ligand. This PD molecule is produced by the tumor. The other subject will talk about the cancer immunotherapy, some lines of cancer immunotherapy. As you know, the cancer is treated by many treatment lines, starting by medicine or surgery, radiotherapy, chemotherapy. And we have another line, fifth line, is called cancer immunotherapy. This is a new line, and some application of this line are routinely given in some cancers, as we will see. So cancer immunotherapy have two types, either non-specific immunotherapy or specific immunotherapy. The non-specific immunotherapy is represented by immunopotentiating agents. These immunopotentiating agents aim as immune modulation to strengthen the immune response against the tumor. So they are non-specific and may be used for any type of tumor. And this mechanism of immunopotentiation might be modifying the co-stimulatory signal. We modified the co-stimulatory signal to enhance the immune response. Also, we can enhance antigen presenting cell activity, and we can use cytokine therapy, or may use monoclonal antibodies. Certain monoclonal antibodies can be used, or we may use tumor cell vaccines. Not all of these mechanisms are non-specific. Some are might be specific for certain tumors, as we will mention. A variety of immunopotentiating agents are used to enhance anti-tumor immunity. So the immunopotentiating agent example, one example is the BCG. The BCG vaccine is vaccine of tuberculosis, used here to potentiate the action of macrophage anti-helper 1, and it is used in the bladder carcinoma. It is routine line of therapy in bladder carcinoma treatment. We have certain synthetic chemicals might be given to potentiate immunity, or cytokines also, and we have certain line termed lymphokine activated killer cells, LAC cells, and this is another mechanism to potentiate the immune system and to make the immunity able to get rid of the tumor. Most of these agents exert their effect by activating macrophage and natural killer cell, eliciting cytokines or enhanced T cell functions. We have some cytokines used in the potentiation of immune function. In this table show some examples: interferon alpha, beta, gamma, interleukin 2, and tumor necrosis factor alpha. And every cytokine has certain mechanism. Interferon alpha, beta, and gamma increase the expression of MHC class 1 by the tumor. Interferon gamma increase MHC also, and activation of the macrophage and natural killer cell and cytotoxic lymphocyte. Interleukin 2 is the growth hormone of lymphocyte. It's called lead to T cell proliferation, activation of natural killer cell. Tumor necrosis alpha also activate macrophage and lymphocyte. Another way to potentiate the immunity is by transfection. It's potentiation of the co-stimulatory signal by transfection of B7. B7 is part of the co-stimulatory signal in cytotoxic T lymphocyte. B7 binds to CD28. So when we transfect B7 gene to the tumor cell will enhance and potentiate the co-stimulatory signal, and thus enhance and potentiate the priming of T lymphocyte. Other ways is transfection with GM-CSF. We transfect the gene of GM-CSF. GM-CSF lead to increased number of dendritic cells around the tumor cell, and this abundance of dendritic cells will lead to wide activation of different type of T lymphocytes, starting by T helper and T cytotoxic, and the cross held between T helper and cytotoxic lead to rapid destruction of the tumor. We have another way by targeting the immune evasion mechanism of the tumor by targeting the CTLA4 and PD1. And these CTLA4 and PD1 are the pathway of energy of T lymphocyte used by the tumor. When we target these pathways, when we inhibit these pathways, we will make the T lymphocyte more active in getting rid of the tumor. This is simply done by the antibody, specific antibody against PD or CTLA4. And this table shows some monoclonal antibodies used in routine clinical practice as treatment targets for some tumor. The most important one is HER2 target, and the drug used is the trastuzumab, which is the HER17 in the breast carcinoma. We have the last one, BCL-ABL translocation, inhibited by imatinib, used in CML treatment, and many other examples. The most important two examples are trastuzumab and imatinib. The last slide shows a new method in immunotherapy against tumor, which is the magic bullet, using the antibody bound to toxin or radioactive material. The specific antibody binds the tumor cell, lead to release of the toxin or release of the radioactive material, which lead to killing of the tumor cell. And this is future therapy. And thank you very much, and we are ready for any question on the classroom. See you.